Today we celebrate our 5th structural biology meeting of Biogune. We have a special lecture, and it's a pleasure for me to introduce Professor Venkatraman Ramakrishnan, or Venki, as I think he likes to be called. You all know that he got the Nobel Prize for his work on ribosome structures in 2009. I recommend, particularly to the young students, young postdocs, to read his autobiography, which I found in the Nobel Foundation webpage, which is called From Chidambaram to Cambridge, Alive in Science. I really recommend it. I will only highlight a couple of things, like, well, he was born in this city in India, where he went to high school and undergraduate in India with 1 year in Alberta, because his father was a good biochemist, and his mother, if I remember, was a psychologist, also with a PhD degree, that ended working out with his father. In the 50s, or maybe 60s, his father published, I think, a couple of papers in Nature, and when Venki got his first publication in Science, I think he wrote his father, I think he was proud. I said, well, you work hard, maybe you get a paper in Nature. I think I read that from in your autobiography. Anyway, so he did a high school and undergraduate in India, and undergraduate in physics, and then moved to Ohio to graduate school, where he got a PhD in solid state theory. And after finishing the PhD, he decided to move to biology. And the reason is that he said that hardly a month went by without a major breakthrough in life science, where physics was having a hard time making any fundamental progress, and he didn't want to spend the rest of his life doing tedious theoretical work. So he moved where the action was, it seems to be then biology. So moved to La Jolla, University of Southern California in San Diego, to do a second graduate, to graduate for the second time. And then a postdoc in Yale, and moved to Brookhaven, where he started his independent career. Was professor in Utah and moved to Cambridge in 1999. I think what is inspiring, and I suggest you to read, is that his the driving force, I think, or this is my opinion, for his work, is that he wanted to have a good education and then move to those places where he thought he could answer best the questions he was interested. So his career has been, I think, driven by the questions and where he could solve it better. And he had been moving from many places. Well, without any more delays, I want to thank you, Venki, for coming to Biogune. It is a real pleasure you are here. And I want to thank you, all of you, for coming to this meeting.
Thank you very much. It's a great pleasure to be here. And I want to thank the organizers for inviting me. It's my first time in Bilbao. And also to thank the British Council for helping support my visit here. So I want to talk about a specific problem of the ribosome, which is called decoding. And just to tell you, to introduce you to the problem, I'll show you a little bit about the history of the problem, and then go on to talk about how high-resolution structures of the ribosome have helped us understand some of the mechanisms involved. So the problem is that is a classical problem, which is to take the information in DNA, which is transcribed into RNA, and convert that into protein. And as you know, the intermediary molecule tRNA mediates that translation of information through anticodon-codon interactions. And the surprise when tRNA was discovered was that the amino acid was about 75 angstroms away from the anticodon. And in the meantime, of course, cell biologists had found that newly synthesized proteins were in these blobs on the endoplasmic reticulum. And when these blobs were isolated, it was found that they all consisted of particles which were about 250 angstroms in diameter and consisted of a large and a small subunit. And these particles were called the ribosome. Now, the point is that about several decades, 4 or 5 decades of work on the ribosome led to a picture where we knew that the 30S bound messenger RNA and presented the codon to the anticodon of tRNA, and the L-shaped tRNA had its amino acyl end buried in the peptidyl-transferase center, where a peptide bond was formed and the nascent chain emerged through an exit channel in the 50S. And, but if you wanted to go further to understand decoding, you have to ask, what does a ribosome actually have to do with decoding? Because it seems that decoding is simply a matter of the codon recognizing the anticodon. So it's all base pairing between codon and anticodon. So what does a ribosome have to do with this? So the first thing to notice is that, actually, you know, the colors are, well, it doesn't matter. The first thing you notice is that the genetic code table suggests that often it doesn't matter what the last base is. And in many species of bacteria, there are fewer tRNAs than there are codons. And this led Crick to propose what's called the Wobble Hypothesis, where he suggested that one tRNA, a particular tRNA, could recognize more than one codon, which differed only at the third position. So the Wobble rules suggest that you have to have perfect base pairing at the first 2 positions, but at the third position, certain kinds of mismatches could be tolerated. And this didn't make any sense in terms of base pairing alone, because why should only the third position matter and not the first? You could understand how the middle position might be a little different. So the other issue with the ribosome is that the tRNA selection is very fast and very accurate. These are actually mutually incompatible goals, speed and accuracy, and yet the ribosome manages to be both fast and accurate. And the point is that you have to have a codon in the A site of the ribosome. In the P site, you have a tRNA that holds the nascent peptide chain, and it's now waiting for the new amino acyl tRNA. And in the cell, there's a pool of all possible tRNA EFTU combinations. So tRNAs are brought in by this factor, elongation factor TU, which I'll talk about later in the talk. And so from this pool, the ribosome has to select just the right one that corresponds to the codon in the A site. And so the question is, how does it do that with that kind of speed and that kind of accuracy? So the first person to realize there'd be a limit to the accuracy of any molecular recognition event was actually Linus Pauling. And in fact, he was worried about a translation problem, although it doesn't have to do with the ribosome. It has to do, at the time he didn't know this, but it has to do with tRNA synthetases. And what he was asking was, how does the ribosome distinguish between valine and isoleucine? I mean, not the ribosome, but the translational system. Because he thought that a system that was designed for valine could easily discriminate against isoleucine, because maybe the valine binding pocket would be so small that it would exclude this extra methyl group. But he didn't understand why a system that could take isoleucine would reject valine accurately, because the only additional interactions would be from this methyl group. And he thought, well, okay, you would have the loss of a few van der Waals interactions, but it shouldn't matter that much. And so he realized that there would be a limit to the accuracy of a system which would be given by the difference in binding energy between the correct and the incorrect substrate. Now that's true if you have simply a binding event and nothing happening, like an antibody-antigen interaction. But if you have an enzyme where the binding leads to a chemical reaction, then the correct distinction is the difference in the free energy of the transition state between the correct and incorrect substrate. And so how does that apply to the ribosome? So if you look at the ribosome, what you see is, from the wobble rules, you can have a GU wobble at the third position, and that's perfectly okay. But that same GU mismatch at the first position would be rejected. Now, you could see on free energy grounds why something that's totally different would be rejected, okay? That would have such a different free energy of base pairing that you could understand the accuracy of protein synthesis. But if you look at a GU versus AU free energy difference, people have done this in solution thermodynamics and shown that it's only about 1 to 4 kilojoules per mole, which means that it would at the most be an accuracy difference of 5 or 10, okay? And yet the error rates, which are better than 1 in 1,000, would require 15 to 20 kilojoules per mole. So the question is, what is happening? One idea of what happens came from a classic paper on antibiotics and the ribosome. And it turns out one of the first ribosomal antibiotics, well, the first ribosomal antibiotic to be discovered was streptomycin. And streptomycin was shown to increase the error rate of translation. And it was also known that streptomycin binds to the small ribosomal subunit. And so Davies, Gilbert, and Gorini proposed that the ribosome had a decoding site where codon-anticodon base pairing was somehow checked, okay? So then what happened is that people started getting mutations. So there was streptomycin resistance mutations in this protein called S12, which made ribosomes more accurate, okay? So they lowered the error rate. And there were other second site suppressor mutations in S4 and S5, which increased the error rate. So you have an antibiotic that increased the error rate, mutations that increased the error rate, and another mutation that, other classes of mutations that lowered the error rate. And in the absence of a structure for the ribosome, nobody knew where all these mutations were. So it was natural to assume that all these mutations were perhaps around the codon-anticodon base pair and were somehow either strengthening the recognition or lowering or inhibiting the recognition, and thereby increasing or decreasing the error rate. And so people like Eigen and Potapov suggested that the ribosome recognizes the geometry of its substrate. In this case, the substrate is the base pair between codon and anticodon. In just the same way that enzymes can distinguish between a true substrate and a slightly different substrate, okay? Enzymes are often very specific. So this idea, though, very quickly lost favor, because although the structure of the ribosome wasn't known, so we didn't know where all those mutations were, the structure of tRNA has been known, well, the secondary structure since the 60s, and the atomic structure since the mid-70s. And so we know that the codon-anticodon interactions are here, the amino acids here. So it was a big surprise when a mutation in tryptophaneal tRNA that allowed the tRNA to read through a stop codon was discovered and shown to be quite far away from the anticodon. So this didn't seem to make any sense in terms of recognizing what's going on over here. So the other interesting thing is then other things happened. For instance, a cross-link between 8 and 13, which is like an inter-strand crosslink, was discovered, and that reversed the effect of this Hearst suppressor mutation. And other mutations like the Smith and Yaris base pair here, or this triple here, if any of these bases were mutated, it would also lower the accuracy. So you had a whole class of mutations far away from the anticodon, which affected accuracy. And incidentally, in one of the 2 tRNA structures that came out, Aaron Klug's article suggested that maybe this base pair switched to forming a base pair with 45, so this was like a little hinge region, and it seemed to occur right in the middle of all of this. But it turns out actually it's not a hinge in the way Aaron thought, but he was sort of close to the mark. So because of this, a different idea was proposed, which is called kinetic proofreading. Now, in kinetic proofreading, what happens is that the ribosome is waiting for the new amino acyl tRNA, which is brought in by EFTU. It binds, and it can bind reversibly. And if it is a correct tRNA, it somehow triggers GTP hydrolysis in EFTU, and the GDP form of EFTU has a very different conformation, and so it dissociates from the ribosome, and then the tRNA swings into the peptidyl transferase center, and then peptide bond formation can occur. And the point is that there are 2 places where you can have dissociation. For instance, you could have a dissociation here, or you could have a dissociation here. And these 2 steps are separated by an essentially irreversible step of GTP hydrolysis. So if you work out the kinetics, if you reach equilibrium at both steps, then the maximum selectivity would be the product of these 2 selectivities. Of course, if you don't reach equilibrium, it'll be less than that. But nevertheless, you'll get a product of what you would get in a single step. And so people at the time thought that the free energy difference from base pairing alone was 1 in 100 rather than 1 in 5 or so. And so they thought, okay, you have 1 in 100 here, 1 in 100 here, and you would get a factor of 10,000, which is more than enough to explain accuracy. Of course, there are several things wrong. The first thing is that we know that the difference between GU and AU is not 1 in 100. It's actually more like 1 in 5 to 1 in 10. The other thing is no one, although this idea has been in textbooks since the mid-70s, no one thought to look at this simple case where this is a cognate phi on its codon, and this is an incorrect arginine tRNA on a serine codon. And actually, this would have greater base pairing stability than the correct combination, okay? It's just that the wrong base pair is in a forbidden location. It's in the first position rather than in the wobble position. And so even if you had proofreading, this should dissociate more slowly than this. And so you should still the wrong one should be selected better. And yet the ribosome has no problem in excluding these incorrect base pairs. So the first step in understanding this came from Marina Rodnina's work on the kinetics of this. And that is what she and her colleagues found is that not only are the dissociation rates different for the incorrect tRNA, and they aren't that different as we know, but the main thing she found was that the forward rates, especially of GTP hydrolysis, was much greater for the correct tRNA than for the incorrect tRNA. So the explanation that they came up with, which turns out to be correct, at least as far as structural data show, is that the correct tRNA somehow induces a conformational change in the ribosome that leads to GTP hydrolysis. And so that's what they proposed. So in 2000, we had solved the atomic structure of the 30S subunit. And so one of the first things we wanted to ask is, how does the structure change when there's a codon-anticodon interaction in the decoding center. And so what we did was take crystals of the 30S subunit and soak in mimics of the tRNA anticodon stem loop and of a codon, of an oligonucleotide corresponding to the codon. And this is basically what we saw. So here is the decoding center, and in purple is the codon, and in red are shown 3 universally conserved nucleotides. And if you see what happens when tRNA binds, what you see is that when the tRNA binds, there's a global domain closure around the tRNA substrate. And if you zoom in on the decoding center, you see what causes or what induces that global change. The global change is actually being induced because these 3 nucleotides they're induced to change their conformation and make interactions with the minor groove between the codon and anticodon. And if you look at the details of the minor groove, what you see is that at the first 2 positions, the ribosome makes tight interactions with the minor groove. But at the 3rd position, it's perfectly happy to tolerate a GU wobble mismatch, as is shown here. Now, the point about minor grooves of nucleic acids is that the shape of the minor groove depends on the geometry of the base pairing. And it turns out that all Watson-Crick base pairs, of course, even in the original Watson and Crick paper, it was known that they were isosteric, that is, that all possible combinations had the same shape. And Siemens and Rich showed that not only do they have the same shape, but they have hydrogen bonding acceptors in exactly the same location, regardless of what the bases are. And so this is a way of recognizing Watson-Crick geometry without depending on the sequence, that is, doing it in a sequence-independent way. And of course, DNA and RNA polymerases also have to do exactly the same thing. And it turns out that DNA and RNA polymerases also inspect the minor groove between the template and the transcript strand and ensure Watson-Crick pairing in that way. But what the ribosome shows is that you can do that minor groove recognition entirely using RNA. You don't need protein amino acids to do that. And this suggests that this kind of accuracy could have evolved, probably did evolve, even before there was a protein world, because presumably the RNA world, or the RNA peptide world, if you will, had to evolve sufficient complexity to evolve something like a ribosome, which would then, allow a transition to the protein RNA world that we have today. So we have the recognition at the minor groove, but what about all those other mutations first in the ribosome and then in the tRNA? Well, I'll talk about the ribosome mutations first. And so here's S4 and S5, and those mutations all occur at the interface between S4 and S5. And this is S12, and the mutations occur somewhere over here. And this 3-ring structure is streptomycin, which also binds right at this interface. So at first sight, it doesn't make sense because they're all over the place, and certainly not around the codon-anticodon base pair. But if you remember that domain closure that I showed you, I can show it to you again from the point of view of this cartoon, and that's what this looks like. So when the tRNA binds, there's a domain movement which allows it to close around the tRNA substrates. And suddenly, things start to make sense. For instance, when the tRNA substrate binds, it induces this domain closure, and that results in disrupting interactions between S4 and S5. And it also results in formation of new contacts between S12 and ribosomal RNA. And it turns out streptomycin would actually make more contacts with this closed form, and so it would stabilize the closed form. So suddenly, you realize that anything that makes the domain closure easier allows the tRNA actually lowers the error rate, and anything that makes it more difficult increases the accuracy. Sorry, anything that makes the domain closure easier makes the ribosome more error prone, and anything that makes it more difficult makes it more accurate. So this led us to suggest that this domain closure is actually required for tRNA selection. And if you make it more difficult, then the incorrect tRNA will have an even harder time getting accepted, and so you'll have higher accuracy. And if you make it easier, then you're increasing the probability that the incorrect tRNAs will occasionally be selected. So the point is, why would a domain closure in the 30S lead to tRNA selection? Well, we had an idea of why this would be from cryo-EM studies on EFTU with the ribosome. And this is how tRNAs are basically selected. They're brought in by EFTU, and then GTP hydrolysis takes place, and then the tRNA has to swing in to the peptidyltransferase center. And so if you look at cryo-EM structures of EFTU with the ribosome and look at our domain closure in light of their structures, what you could see is that the EFTU, which is now, of course, cryo-EM is now at between 6 and 7 angstroms on the EFTU complex. You can see that the domain closure would move the shoulder of the 30S subunit towards EFTU and tRNA. And so what we proposed was that the domain closure is somehow triggering the GTP hydrolysis on EFTU by sort of inducing the transition state. The transition state involves this closed structure of the ribosome. So, of course, cryo-EM has gone to 6 to 7 angstroms resolution, but if we wanted to understand the details of all of the interactions at the active site and of the nature of the interactions here, we wanted to try and get a high-resolution structure to see what's happening. So that took us quite a long time. We started in 2001 when the best structure of the ribosome was at about 5.5 angstroms. And we wanted to get a substrate-bound structure that would diffract to high resolution. And so it was a series of steps, first to make high-quality ribosomes and then scale them up. And then 2 postdocs, Maria Selmer and Christine Dunham, tried to crystallize an EFG complex. And when they obtained crystals, the crystals first went to 6 and then 4.5, and eventually went to beyond 3 angstroms resolution when all of the people in the lab basically helped Maria and Christine to build and refine this structure. And that structure is shown here, and it's a complete high-resolution structure of the 70S ribosome with mRNA and tRNAs. Now, this structure has been useful for lots of things, including, for instance, understanding the details of how release factors recognize the ribosome, and other people have used it for antibiotics and EFP and so on. So, however, it turned out not to be useful for either EFG or EFTU. In fact, even though we started with an EFG complex, there was no EFG in the crystals, okay? And the reason for that is that in this crystal form, as well as in all previous 70S crystal forms, including the low-resolution form and an E. coli empty ribosome, all of which were in different space groups, they all had a common feature, which is that this protein L9 stuck out of the ribosome and bound to a neighboring ribosome in the crystal. And it did it in such a way that it would prevent the binding of GTPase factors. And even if you started with GTPase factors, the equilibrium for crystallization would drive out the factor. And so Maria Selmer and Albert Weichselbaumer decided to delete the gene for L9 from thermus. So they went ahead and created a thermus strain which lacked ribosomal protein L9, lacked the gene for L9. And then using that strain, John Weir and Frank Murphy obtained initial hits for an EFTU tRNA complex, but they both left the lab for various reasons. And nothing happened for a while until Yonghui Gao used that L9 deletion strain to crystallize and solve an EFG complex. And this motivated Martin Schmeing and Rebecca Voorhees and Ann Kelly to start with these initial hits, but it still took quite a lot of optimization, over 1,200 trays. Each tray is about 96 drops, so you can get an idea of how hard they had to work. And they eventually ended up with a structure of an EFTU tRNA complex in the state after GTP hydrolysis, but trapped with chiromycin, so it prevents EFTU release. Now, since then, we have also solved the structure before GTP hydrolysis using a non-hydrolyzable GTP analog at somewhat higher resolution, at about 3.1 angstroms. So I'm going to, for the rest of the talk, I'm going to just tell you what we've learned from these structures. Well, the first thing is, of course, the tRNA is not in the A site of the 50S, but that's been known for a very long time, first from chemical footprinting studies, which showed that the EFTU tRNA binds in a different place on the 50S subunit, although the site on the 30S subunit is the same, which is the A site, so it's called an AT tRNA. And then cryo-EM studies confirmed that original finding, but they also showed subsequent cryo-EM studies showed that there's a bend in the anticodon stem of the tRNA, and this bend was characterized better in subsequent higher resolution studies. And of course, people in Bilbao will recognize this name here. So what we've learned now is the sort of cause of the bend and what that means. So here is the A site tRNA as it would look after accommodation. And this is the EFTU bound form, which is highly distorted. And if you look at where the distortion is, it's right in the anticodon stem, and it's not a kink. It's actually an untwisting of the helix, which results in a broadening at the same time of the helix. And as you know from topology of double helical nucleic acid, if you untwist something, you can increase the rise of the nucleic acid and thereby induce a bend. And if you want to have an idea of how this works, you can take a telephone coil that's helical and just try and untwist the helix, and you'll see that it's taken up as the bend. So anyway, that's what it looks like. That's the result why the tRNA goes off in a different direction. And you can see that there's a widening here of the groove of the tRNA. And one of the interesting things is that the base pair, which when mutated causes an increase in the error rate, is right there at the position of the bend. And so you can see that if you widen this groove, you would break this interaction and thereby make the tRNA more easy to distort if you mutate it. Okay, so if you mutate that interaction, then you don't have to pay the penalty of breaking that interaction when you bend it. So again, the same principle holds, that anything that lowers the energy of distorting the tRNA, which is part of this transition state, makes it easier for the tRNA to be accepted and thereby increases the error rate. So the other distortion happens at the D stem, and there's a movement of about 5 angstroms at the D stem. And if you look at what's happening, you see that the Hearst suppressor is here, right at the sign of this distortion. Now, when we solved our first structure, we couldn't tell why the Hearst suppressor would cause a change in the accuracy, because in the Hearst suppressor, what you're going to do is replace a GU pair with an AU pair. So the interaction, if anything, would be stronger in the Hearst suppressor. And so we didn't understand why the Hearst suppressor would cause a lowering of accuracy. I'll come to that in a minute. But you can understand why cross-linking 8 and 13, you see these strands move quite a bit. And so if you cross-link them, you would inhibit that movement, and so you would make tRNA distortion more difficult, and so it increase the accuracy and, of course, compensate for the lowering of the accuracy in the Hearst suppressor. And this is an interesting pair of residues, because this is where a fluorescent dye, proflavin, was introduced for the kinetic studies. And you can see why it's a good reporter for decoding, because it's a place where there's local conformational changes when EFTU binds to the tRNA. And finally, you can see how disrupting this base pair, this base triple, would make it easier for the distortion to occur. So again, all of those tRNA mutations can be explained using a simple single principle, which is that anything that would make it easier to distort would actually lower the accuracy. So it's just like the domain closure in the 30S subunit, the same idea. Now, what about the Hearst suppressor? So recently we've done a structure of the Hearst suppressor, which is this tryptophanol tRNA with a G24A mutation. Now, it turns out that this is the tryptophanol tRNA trip, and this is the Hearst suppressor, okay? Now, what happens in the Hearst suppressor is that in the distorted form of the tRNA, not in the undistorted form, but in the form with EFTU on the ribosome, you have a slightly stronger base pair between AU versus GU, and you have this additional interaction of A24 with G44. Now, these bases are far away in the undistorted form of the tRNA, but they're brought closer together in the distorted form. So what the Hearst suppressor is doing is forming an additional interaction that strengthens the stabilizes the distorted form. And so that's why you can see why that's why the Hearst suppressor stabilizes the distorted form and increases the error rate, or allows the read-through. And in fact, if you look at the efficiency of read-through, it's pretty much consistent with the kind of energy gains you would get from these additional interactions. So for the last part of the talk, I'll talk about EFTU and catalysis. So here's EFTU, and when you compare it off and on the ribosome, what you see is there are some changes here, but there's a significant change in this loop of EFTU, which interacts with the small ribosomal subunit. And if you look at that interaction, what you see is this loop is almost over 99% conserved in bacteria. And there's no reason why the tip of a loop should be highly conserved unless it's actually doing something important. And you can see that what it's doing is stabilizing interactions with the 30S subunit using highly conserved nucleotides. The other thing you can see at the edge of this picture is there's a stacking interaction between C75, which is the penultimate base of tRNA, and a base, a purine of ribosomal RNA in the 30S subunit. And if you look at it in a broader picture, what you see is normally the tRNA 3' end is in this conformation in EFTU, and when it's bound to the ribosome, this 3' end gets pulled towards the small subunit and is stabilized by this stacking interaction with the 16S RNA of the small subunit. Now, the consequence of this is that it moves by about 6 angstroms, and this 3' end normally makes an interaction with this switch 1 of EFTU, okay? Now, the first structure we solved was after GTP hydrolysis, and we couldn't see switch 1 at all because it's disordered in the GDP form. And so we concluded that because the ribosome is pulling away the 3' end, and due to this stabilizing interaction, it's breaking these interactions with the switch 1 helix, and so switch 1 becomes disordered. And the reason for that was that it was a dogma in the EFTU field that switch 1 was protecting the gamma phosphate of GTP. It was like a hydrophobic gate. And when switch 1 became disordered, it would allow water to get access to the gamma phosphate of the GTP and allow hydrolysis. And so we thought that ribosome binding by pulling away the 3' end is destabilizing switch 1 and thereby triggering GTP hydrolysis. But actually, now that we've done the GTP structure, we know that's not true. And again, it's one of these things where people compared the EFTU GTP structure with the GDP structure. They saw a difference, and they simply said, oh, it's a hydrophobic gate, without actually asking, is there enough space for a water molecule to go there when switch 1 is ordered? Okay, so they never asked that very simple question. And so you can see that you can be misled by structure if you're not careful. So what's happening at the catalytic site? So I thought I would wake you up a little with this slide. This is a guy who defected from Bulgaria and became a journalist for the BBC. And this was during the Cold War. And because he was a defector, the Soviet bloc hated him because he knew the inside story of what was going on in the Soviet bloc. And so the poor guy was standing at a bus stop in London one day, and some guy who later turned out to be a spy in the Bulgarian embassy, stabbed him with an umbrella, okay? And 4 days later, he died. And the reason he died was because he was injected with the deadly poison ricin, okay? So the umbrella had a pellet gun, which injected a pellet of ricin into him. Now, ricin is a toxin that depurinates a single adenine in 23S in large ribosomal RNA, large subunit ribosomal RNA. And a single molecule of ricin can inactivate all of the ribosomes in the cell. So the question is, what does ricin do? Well, what ricin does is, I told you, it depurinates an adenine in this loop of 23S RNA called the SRL, which stands for the sarsen-ricin loop. And the other toxin, alpha-sarsen, actually cleaves a nucleotide at the sarsen-ricin loop. So you can see, and this was already known from cryo-EM structures, that the sarsen-ricin loop is closest to the GTP molecule in EFTU. So what is the role of the sarsen-ricin loop? Well, here is EFTU. Here's GDPCP, which is the GTP analog we used. And this is a structure by Paul Nissen's group of the isolated ternary complex of EFTU with tRNA with GDPCP. And this histidine is universally conserved in both eukaryotes and in prokaryotes and all translational GTPases. So IF2, EFTU, EFG, and RF3. So you can see that this histidine is pointed away from the gamma phosphate. And by the way, if you mutate the histidine, you get a big change in the rate of hydrolysis of GTP. So when it's bound to the ribosome, the histidine has changed conformation so that it's now coordinating a water molecule that's poised to attack the gamma phosphate. But notice that switch 1 is still ordered. It's not disordered, okay? And so this means that it doesn't have to move or become disordered a lot to allow water molecule in. Actually, there's plenty of space here for the water molecule to come in. It's just that if the histidine is not there coordinating the water molecule, then you don't have a coordinated water molecule for attack, and so you don't get hydrolysis, okay? The water molecule is not stable there, and there's no reason for it to be positioned properly for inline attack. So then you have to ask, why is the histidine, if this is a favorable position, why is the histidine in this position to coordinate the water molecule? And that's where the sarsen-ricin loop comes in. You can see that A2662, in fact, sarsen cleaves right here. One of the non-bridging phosphate oxygens is bonded to the histidine and holds it in just the right orientation to be coordinated to the water molecule for attack. So what the all these induced conformational changes do is position the EFTU so that the catalytic, well, the activating histidine is in just the right orientation, placed in just the right orientation with the sarsen-ricin loop. And we can also sense from this how other GTPase factors work. So EFG, IF2, they all have this catalytic histidine. They have a homologous GTPase domain. And the point is that although they're slightly different, they all bind to different states of the ribosome. And as you know, the ribosome is highly dynamic, so the orientation between the subunits isn't exactly the same in the different states. And so each factor will bind to the ribosome correctly only for its particular state, okay? And by correctly, I mean it'll be positioned in a stable way with the histidine at the sarsen-ricin loop. And so it explains also, in a way, the specificity of these factors. So finally, this is a summary, which is you have the inactive histidine on ribosome binding. The sarsen-ricin loop stabilizes an active form, which results in hydrolysis of the GTP. And then after GTP hydrolysis and phosphate release, so you need phosphate release to destabilize the switch 1. Then switch 1 becomes disordered, and then EFTU adopts a completely different conformation, okay? And so here's the whole sequence of events that I have now described in my talk. So first, EFTU binds, and the tRNA can sample a variety of conformations, but the distorted form is not really stable, so it would be a very short-lived conformation normally. But if you have codon-anticodon interactions which are correct, then what will happen is you will get this additional sort of clamping of the codon-anticodon base pairing by these minor groove interactions, and that will stabilize the distorted form of EFTU tRNA and move the shoulder up towards EFTU and stabilize a distorted form and also distorted form of the 3' end of the tRNA. And all of that will position the sarsen-ricin loop to activate the histidine. And then after GTP hydrolysis, the phosphate will leave and the histidine will adopt a different conformation that's inactive. And then once switch 1 is disordered, EFTU changes conformation. So now it's broken most of its contacts with the ribosome. And it also has a lower affinity for tRNA. So it will leave, and then there's nothing to hold the tRNA in a distorted form. And so the tRNA will relax into the peptidyl-transferase center because it's still being tightly held at this decoding center. And then that will allow a peptide bond to be formed. So I've told you a lot of this using a lot of words, but I thought I would end with a movie that my postdoc Martin Schmeng and student Rebecca Voorhees made. And they thought the movie would be much better than any words that I could have used to describe this thing, and you'd find it easier to remember. And then they had the idea that maybe if they used songs which had words that were relevant to what was happening, for instance, a tRNA deciding whether to bind or not to bind, and so on, then you would really remember, especially those of you who are under 40 years old. So here's the movie of the process as we see it today.
Well, come on and let me know. Should I stay or should I go? Should I stay or should I go now? Changes. Don't wanna be a richer one. Get into the groove, boy. You got to prove you're to me. Get up on your feet, yeah. Step to the beat, boy. What will it be? Music can be such a revelation. There is a season, turn, turn, turn.
you deep inside. There's a secret garden she had.
I'm leaving on a jet plane. I don't know when I'll be back again. Oh, babe, I hate to go. Jack, don't you come back no more, no more, no more, no more. Hit the road, Jack, and don't you come back no more. We are the champions. No time for losers, 'cause we are the champions of the world.
Thank you very much.
I have one question regarding the effect of the mutations on the tRNA in the selection. Because you have two steps of selection. The first one is to get the distorted one, and the second one to recover the original structure in accommodation. So could you comment in the, whether you suspect or you know that the effects of those mutations are different in both of the steps?
I don't know if there are any mutations that are specific for the second step. So the only experiment that I know of is from Rachel Green's lab, where, if you remember, she did kinetics of the Hearst suppressor tRNA, and she found that the GTPase step, GTP hydrolysis rate, was accelerated for the Hearst suppressor, so it was almost the same as wild type, as cognate tRNA. But but in her case, I don't think she actually measured the accommodation rate, to see if that step was affected. My guess is that most of the mutations actually affect the first step and not the second step. And it's not entirely clear. People always argue, and in different buffers you get different accuracies. People argue about what is the relative contribution of accommodation versus the first selection. And I actually think most of the effect is in the first step. And of course, the second step contributes a little bit, but not as much as the GTP selection.
I have a very naive question about non-chemical. I mean, I'm not a biologist, I'm a chemist. So you said that the domain closure actually reports on the speed. So there are other possibilities, other mutations that actually reduce the rate of failure on the incorporation. So my question is then this regulated by the speed of the RNA incorporation, and could be actually slower, and then it would be life would be actually slower? That would work?
Yes, so the streptomycin-resistant mutations, the hyperaccurate mutations of the ribosome, which increase the accuracy by making the domain closure more difficult, they also grow more slowly, okay? So in that sense, there is a relation in that by slowing, they also slow down the process. But they slow down the process by making a step more difficult. And so, the rate even for correct tRNAs is slower than it is for...
But the question is that they live longer?
Well, what do you mean by living longer? I mean...
Yeah, that's... well, I don't know. I mean, it's just that they have less mutations, therefore, in principle, in a cell, they would have less cancer, or... I don't know.
I mean, E. coli...
I understand. Of course.
So there's no apoptosis in E. coli, so I don't know.
I told you I'm not a biologist.
But in the wild, they would be at a competitive disadvantage, and that's why they don't. So in the wild, there's always a compromise between speed and accuracy. Mm-hmm. And, protein synthesis is the least accurate of the central dogma processes. So replication is by far the most accurate, and then transcription is in between, because the same copy of mRNA can make many different copies of the protein. And then in protein synthesis, you need a certain amount of accuracy, but you don't have to have 100% accuracy, because as long as you make enough of the correct enzyme, it might be okay, you know. So, or even if many of the errors would be of similar amino acids, you know. So,
What was the breakthrough that moved from 4.5 angstrom to the 2.8 angstrom in terms of methods, in crystallization methods? That's the first question. The second
yeah. So, you know, the 6 angstroms were very, very small initial crystals, and so 6 to 4.5 was simply improving the size. And then 4.5 to 3 was actually a big surprise to me, but I think it was largely optimization, trying to add, you know, extra divalence, a little bit of dehydration. So we increased the PEG concentration. The crystals were grown in PEG, and we increased the amount of PEG, so they were slightly dehydrated. So I think a lot of those combinations, they're always small steps that result in, you know, and that was actually, you know, we can routinely get sort of 3.2 to 3.5. So even today, that was an unusually good crystal, you know. So.
And the last question is, how many crystal structures have gone in that movie?
Sorry?
How many crystal structures have gone in that movie, in the last movie that you presented?
Oh, I hate to think of that. Well, first of all, there were the EFTU structures, you know, which were, you know, solved many years ago. And then there was the ternary complex structures, which was solved by Paul Nissen and his colleagues. Then there was the empty ribosome structure. Then there was the chiromycin structure. Then the GTP analog structure. And then there was the accommodated structure. So there were a huge number of structures. Yeah.
If I remember well, there are some screenings looking for genes that promote and control tumoral growth that are ribosomal proteins. So are these ribosomal proteins conserved in the bacterial ribosome, and how?
Yeah, so these are all from the eukaryotic ribosome, and some of them don't have a bacterial counterpart, and one or two do though, and I can't explain on the basis of structure why they should, you know, have you know, some effect on tumor growth. And one idea that people in that field have suggested is that maybe these ribosomal proteins have a moonlighting function in some other process, you know, which may be involved in tumor regulation.
My question is about Tyranny structure. I know that, and I think everybody knows that they are all the same, more or less, all Tyranny. But in terms of flexibility, is there any difference between different tRNA? And if yes, then does it matter for the process you described?
So we've done 3 different tRNAs, and they're similar but not identical. So for instance, that close approach between G24 and G43 in the Hearst suppressor would not occur in threonyl tRNA or phenylalanine tRNA, but only occurs in tryptophaneal tRNA. So there's slight differences in the 3-dimensional structures of all these tRNAs. And so the way that they distort is also slightly different between threonyl and tryptophaneal tRNA, okay? So they all distort, but the details of the distortion are slightly different. So they're not identical. Now, the other thing is that when we've done experiments with in vitro transcribed tRNAs, we can't see very good density at all, okay? And the difference is that natural tRNAs all have modifications, okay, which presumably stabilize them in some optimal way. They can't stabilize them too much or they would find it too difficult to distort, but stabilize them in some optimal way. And the third thing is that if you look at Aki-Uhlenbeck studies, tRNAs have evolved so that all of the amino acyl tRNAs have a similar affinity for EFTU, okay? But if you mix and match the amino acid and the tRNA by using some tricks, then the affinities can change by 2 or 3 orders of magnitude. So this means that all of the tRNAs have evolved to have some compromise, of EFTU affinity and of, distortability and binding affinity. So that's my guess, so that the system doesn't have bottlenecks. No matter what the codon is, the rates will be more or less the same to keep the machine going.
Venki, you just mentioned that tRNA is very heavily modified, and I wanted to ask, could you tell more about the role of these modifications? Because
the conventional feeling which we indirectly see is that these modifications add to the stability of tRNA in some optimal way. But if we analyze a structure, we can't, we couldn't have predicted it. So I think it just means that I don't know enough about, how these modifications contribute to stability.
But you mentioned that the dynamics of tRNA and ribosome has a very important role.
I haven't specifically looked at the role of modifications, to be honest.
Because presumably they stabilize the structure of
they
tRNA
must,
to some extent.
and certainly our results with in vitro tRNAs seems to confirm that.